ever seen. And you go from that right down to submicron particles. What Judy's work showed was that larger aggregates disintegrated under regular tillage, and particularly where you had no organic inputs. At the same time, the smaller aggregates resisted this breakdown, so you began to get a separation between macro and micro aggregates. On the basis of all this information, we put together 4 stages of aggregation, and this is taken out of the paper. You start off with 2,000 micrometers, where you've got a solid with pores. You come down to smaller aggregates, where you can see the roots and hyphae holding things together. Now, one of the things that Judy was able to establish was that these fungal hyphae were mycorrhizal. Normal fungi are not particularly involved in holding things together, but the mycorrhiza associated with roots are extremely important. Then as you go down through the sizes, you start looking at bits and pieces of organic matter encrusted with clay, until you get down to less than 0.2 micron sized particles, where there are some organics on the surface. And that's the model that we actually put forward. And it simply says larger aggregates are stabilized by roots and hyphae, smaller aggregates are stabilized by the decomposition products of roots and hyphae, including fauna and bacteria, etc. And when you go into the chemistry of that, it gets enormously complex. You can simplify all this by saying there are microaggregates, say less than 250 microns. There are larger aggregates which are greater than 250 microns. They're very dependent on roots and mycorrhizal hyphae. And finally, we put all this together in a very simplified diagram. Aggregation plotted against wheat fallow, pasture fallow wheat, and so on through to permanent pasture. The roots and the hyphae, temporary binding agents which disappear as soon as you go into too much tillage and you don't have the organic inputs. The other things are there all the time and represent microaggregation. Now I'll stop there and let Judy deliver the goods for you.
When I came to Adelaide to do my PhD in the late 1970s, I was lucky. I was at the right place at the right time. At the Oades Institute and at CSIRO. At the Oades Institute and at CSIRO, there was an active mass of minds, as there is today, all working on the world stage, but all willing to talk to students. 4 people in particular helped me with my project, Malcolm of course, but also Jack Walker, who was in the microbiology department, Sally Smith, and also Kim Bowen, who was with CSIRO. I was lucky with my first experiment. I showed that ryegrass grown for 8 weeks in soil was able to build up aggregates greater than 250 micron at the expense of aggregates 50 to 250 micron. And the aggregates were held together by roots and fungal hyphae, as Malcolm said. Jack Walker identified several types of mycorrhizal fungi in my samples, and this was a breakthrough. I was just happened to be in the right place at the right time. Jack Walker was one person in the world who could identify different types of fungi just by looking through a light microscope. And previously, as Malcolm said, previously it had been thought that fungal hyphae were not important in aggregation because they don't persist in soil, but they weren't thinking of mycorrhizal fungi. And Sally and Dean were already famous for their work on mycorrhizals, and they were very helpful to me on methods and also just learning about these interesting fungi. Yep. And as Malcolm said, this is our model, but our paper came out of, it was really my lit review for the thesis and also some of my results for the thesis. And as Malcolm has said, we showed that there was a hierarchy of aggregation. Aggregates were built up in steps. We said you started off with these small aggregates. So these small aggregates are 0.2 of a micron. And they in turn were held together into these stable aggregates of 2 micron. And here there's microbial debris with clay particles stuck on the outside. And those in turn were bound up into larger aggregates and then larger aggregates again. So there's a hierarchy of aggregation, different levels of aggregation, and each of these has different binding materials. And because these are tiny aggregates, the binding materials have to be tiny. So roots and hyphae wouldn't fit in here. And so you go through these different binding materials up to the roots and hyphae, holding the macro aggregates together. And then a couple of years later, Malcolm proposed the first modification to our model, and he suggested that aggregates were not built up in steps from the smallest to the largest, but macro aggregates formed first, and then stable micro aggregates formed within the macro aggregates. And he said that the roots and hyphae were broken, probably by faunal activity, and became fragmented, and they became coated with mucilage and clay particles, and these became the stable micro aggregates. Elliot used organic carbon in soil. He was working with native grasslands that had only recently been tilled, and he showed that most of the organic carbon released with tillage is from the macro aggregates, from the large aggregates, and so that macro aggregates turn over much faster than micro aggregates. And the organic carbon in the macro aggregates is younger and more labile and less decomposed than that in the micro aggregates. So that they're really the roots and the hyphae. And then the larger the aggregate, the higher the concentration of organic carbon. Because there's a hierarchy of aggregates, there has to be a hierarchy of pores. So Elliot and Coleman defined these 4 sizes of pores, and they said, well, there are biopores, which they're the pores that are formed by roots and by, say, earthworms. And there are also cracks that are formed in the soil by wetting and drying of the soil. Then the next stage, so these ones are they're greater than 600 micron. And then there are macropores between macro aggregates. So if you look at this picture here, these are 4 macro aggregates, and there's space between here. and they're drained at field capacity, so they're going to be 100 micron or so. So these ones here are mostly drained because they're quite large. These ones are drained at field capacity. But then there are also pores within the macro aggregates. So there are pores within this, but between the micro aggregates. And then these micro aggregates are the darker bits. And there are pores within those as well. And then Tony Dexter talked about the pore exclusion principle. And he said an aggregate can't have pores in it that are almost the size of an aggregate. So here, this is a macro aggregate. It can't have a pore as big as itself. And the large aggregates contain the pores both between the small aggregates and pores within the small aggregates. So the large aggregates have a higher porosity. And because of all of this, it means that the smaller aggregates are more dense. They've got smaller pores, fewer pores, smaller pores. And that they've got higher internal strength because the pores are smaller in the smaller aggregates, so the particles are closer together, and so there's more chance of those particles forming into aggregates. And then Elliott and Coleman said within these 4 levels of pores that animals or fauna of different, or organisms of different sizes live in different pores. So these are the large pores, the biopores and the cracks, and they contain fauna such as the microarthropods, which are organisms. They could be small beetles or organisms with a hard an exoskeleton. Then these ones are the next. They live in the next size pore down. They're smaller organisms, so they're actually living in pores between macro aggregates. And then the next size pores have these organisms, and they're smaller again, and so they're living in pores between the micro aggregates, but within the macro aggregates. If you remember back to this one, these were between the macro aggregates, and these are within the macro aggregates. And then the smaller pores have the bacteria, and they're living within the micro aggregates. These animals, or these organisms, all need room to be there, but they also need room to move, and for nutrients and water, they need access to those. So the pores have to be bigger than the organism, but they need to be small enough to keep out the predators. So, for example, bacteria, which live in these tiny pores, they are protected from, say, protozoa in particular, who are predators on bacteria. So they're protected in the pores. And Malcolm and Angela showed that not all soils show this hierarchy of aggregation. So they looked at 3 different soil types, and they subjected them to different amounts of physical energy. Earlier than that, people had often used chemicals to break bonds, but Malcolm and Angela used energy. So they subjected their soils to different amounts of physical energy, from gentle disturbance to vigorous disturbance, which was with ultrasound. And they showed that it was only the soils that were stabilised by organic matter that showed that broke down in stages. And alphesols, they're the soils, or red brown earths, fit into this one here, and they have illite and kaolinite as the clay. Mollisols have smectites, And but Malcolm and Angela found another step when they applied in different amounts of energy. They had found these aggregates of about 90 to 250 micron, and inside those aggregates they could see plant remains. By the time that they got down to this size, the plant remains were not organised. Whereas with the oxides, Malcolm mentioned, they're not stabilised by organic matter, and so they didn't break down in steps. They're very stable, and the only way that they could be broken was with ultrasound. And then another student of Malcolm's, Golden, that developed a model of turnover of microaggregates. So they took several soil types, removed the light fraction, which is the particular organic matter that's not attached to soil. They floated that off in heavy liquid, and they then applied ultrasound for 5 minutes, and were able to collect these different